Hi, it's Mr. Mazurkewitz, and in this video I'm going to be talking to you guys about the process of mitosis. And mitosis is just the way that we're going to take cells and create more cells from them. So if you're a single-celled organism like this guy here, this is how you're going to reproduce. It would be a type of asexual reproduction. You're just going to make a genetic copy of yourself, and it's just going to be how your main way of reproduction. In the case of multicellular organisms like ourselves, other animals, plants, this is going to be a great way to produce more cells in the event that maybe we have damaged tissues, so we have to replace the ones that were lost. We'll use mitosis, or in the event of growth. So here's a picture of me in the first grade. I know what you're already thinking. I used to be so cute. What happened? Life happened. But what has happened to me since then? Why am I so much bigger? It's not that my cells got bigger. It's that since then I've been producing more cells. We all start off as one single cell, and it's through the process of mitosis that we've created the hundreds of trillions of different cells that make us up today. So our lesson essential question today is going to be focused on mitosis, and we should know first of all the different phases of mitosis. There are going to be 4 parts to mitosis that we're going to go through. You should know what they are, and also what occurs in the cell at each one of these stages. So not only knowing their names, but knowing what's happening at each of these 4 stages of mitosis. Before we do that, I wanted to take a quick detour and talk about our good friends, the prokaryotes. We've talked about them in the past. These are guys like bacteria. They are not going to do a process of mitosis, but they use something else called binary fission, which is pretty much the same thing, just a lot simpler. So if you remember prokaryotes, they don't have a nucleus to deal with, so they don't have to worry about that getting in the way. And they also don't have as much DNA. They only usually have one single circular chromosome. They don't have 46 like in us, where they're going to have a lot more going on. So the process of binary fission is a quick, very simple way that bacteria are going to reproduce. They're going to take their circular DNA chromosome, they're going to, through the process of DNA replication, make a copy of it, and then simply just divide so that each new cell gets a copy of that DNA. This is a very efficient way that bacteria reproduce, and it only takes about 20 minutes, so it must have worked. They've been around for billions of years. So again, binary fission, to recap, all they're doing is making a copy of their DNA and then going to divide, giving each new cell a copy of that DNA. It's a very, very simple, quick way to do it. For other eukaryotes, like ourselves and plants, we have to use the process of mitosis. So there's going to be 4 major parts of mitosis. There's going to be prophase, metaphase, anaphase, and telophase. So if we're to take a look at what mitosis really is, it's really just the division of the nucleus. A lot of people think it's how the whole cell divides, but really mitosis is how we're going to take the nucleus of a eukaryote and divide it so we end up with 2 nuclei that are genetically identical. So we take a look at a plant. Here are all the various stages. So here's a plant cell to begin with. You can see its nucleus has been stained, and through the process of prophase, shown here, metaphase, finally anaphase and telophase, we end up with a single cell. So we haven't divided the cell yet with 2 nuclei. There will be a final step here that sometimes overlaps with telophase that we'll talk about, of where we finally divide the cell into 2. So we're going to take a look at each one of these steps and talk about what's going on inside the cell through them. So before we get into prophase, the first step of metaphase, I wanted to quickly recap the other parts of what is known as the cell cycle. So the cell cycle, this is a picture shown here. It's just this cycle, a circle of what's going to happen in a cell in order for it to keep pumping out a new cell each time, how we can divide. So the whole first part, the majority of the cell cycle, is a phase known as interphase. And interphase consists of 3 parts. There's going to be G1, S, and G2. So G1 is going to be this period of growth where the cells are making organelles and proteins. It's going to increase its size. Then in the S phase, the synthesis phase, this is where all the DNA is going to be copied, the most crucial part. And then the cell is going to enter what is known as G2, which is another period of growth. And then finally, now the cell is ready to hit this last final stage of the cell cycle, the shortest but most crucial part, which is known as the M phase, or mitosis. Before we talk about mitosis, I also wanted to mention that cells, you might think to yourself, what if a cell doesn't want to divide? Well, it can enter into what is known as G0. So we can have a G0 phase where the cell is just kind of hanging off to the side, being a cell, not really getting ready to divide. But then when it wants to divide, or needs to, it will then enter back into G1 and continue that interphase and go into mitosis. So again, interphase is the majority of it, and that is just a period of growth, DNA duplication, and then further growth. So once we head into mitosis, the first part is what we call prophase. So prophase, a few things is going to happen. The first thing is we're going to take all what is called the chromatin, which is just all the DNA unraveled. It's kind of unorganized, you see in this picture here. And we're going to condense them into these structures called chromosomes, which you might be more familiar with. This is important because if I told you to sort all that chromatin in that picture into two halves of the cell where you're going to separate them, it'd take a really long time. It'd be very hard. So what our cells do is, the first thing we're going to do in prophase is take all that chromatin and condense it into chromosomes. So here's a picture of a chromosome. It's got an X shape, and each half of that X is what we call a sister chromatid with a D, chromatid. And these are genetic copies of each other. So during that S phase, this is where we copied all that DNA. And this whole process is about taking that X and breaking it off. So one half goes one way, and the other half, the copy, goes the other way. So again, a crucial part, we're going to take that chromatin and condense it or pack it up into these chromosomes. A few other things are going to take place that are crucial. The other thing is, it's still in prophase, the nucleus or the nuclear membrane is going to break down. The reason for that is we want to get to the DNA. The nucleus secures the DNA, so we want to get rid of that nucleus. And also we're going to get these things forming called mitotic spindle fibers, which are kind of like microtubules. These things are going to form and they're going to attach to a chromosome. So if we're going to have our cell here with our chromosomes, so I'm just going to draw a couple here, what we're going to form are these mitotic spindle fibers, which are just these fibers or these microtubules that'll connect onto those sister chromatids so that we can organize them, we can move them where we want, and most importantly, later on, this is how we're going to grab onto them and pull those sister chromatids apart. So all this happens in just prophase, that first part of mitosis. After prophase is complete, we're then ready for the next stage, so the M in PMAT, which is metaphase. Metaphase, the way I remember this is I just think of the M in metaphase reminds me of the word middle. So really what's going to happen is the chromosomes are going to line up across the middle of the cell along what we call the metaphase plate. The metaphase plate is just this imaginary line where all the chromosomes are going to line up. In our cells, imagine 46 of these guys all lined up across the center. The reason for this is important, is because this is how we're going to organize to make sure that half of the chromosomes go one way and half of the chromosomes go the other. We don't want to get this mix and match of chromosomes where things are unequal. So this is just a great way to keep things organized by lining them up in the middle. Once we do that, we're going to move on to the A of PMAT, which is anaphase. So in anaphase, the sister chromatids are now going to get pulled apart to opposite sides of the cell. So remember those spindle fibers that were connected, they're going to shorten, and as they shorten, they're going to pull one chromatid one way and the other sister chromatid the other. And this is important because remember, those guys were copies. We don't want them in the same cell. So we're going to make sure that half of the genetic information goes to the left, half of it goes into the right. Once we do that, we are now going to head into our last part of PMAT, the T, which is called telophase. Telophase is pretty much the direct opposite of prophase. So everything we did during that prophase, we're going to undo. So the spindle fibers, for example, those microtubules that we formed, those things are going to disassemble. We're going to get rid of those fibers. We don't need them anymore. They did their job. Those nuclear envelopes, the nucleus that we broke down, we're going to reform them. The only difference now is now we need two. So the nuclear envelope is going to form around the separated chromosomes, now forming two nuclei. We'll have one nucleus around the ones that went to the left, one nucleus around the ones that went to the right. And then finally, those chromosomes that we had packed up, those are going to unravel back into that crazy spaghetti-like that we saw before called chromatin. So we're going to unravel them because we don't really need them organized anymore. What you notice now, though, if we look at this picture, is now we have one big giant cell with two nuclei. So you should probably say, hey, wait a second, I still have one cell. This isn't right. That's because, like I said before, there is one more step. It's not part of mitosis, but it does kind of overlap. So while telophase is going on, the last thing to happen is a process known as cytokinesis, which sounds really cool. And all cytokinesis is the division of the cytoplasm, excuse me. So we're going to take that cell, and it's going to start to pinch off until we finally separate the one cell into two what we call genetically identical daughter cells. And this can happen one of two ways. So in the case of animal cells, you remember that animal cells just have a cell membrane. So it's very easy for it to pinch in. So you can see in this picture taken with an electron microscope, you can see the actual cell pinching in down the center, and eventually we're going to pinch off enough where the cell will separate into two different cells. So that's how we're going to create our two daughter cells. Now if you're thinking, what about plant cells? What do they have on the outside of their cell membrane that won't allow them to pinch like this? They have a rigid cell wall. So here's a picture of a plant cell, and it might be hard to see, but what we have going on, let me get my red here. So what we have developing in the middle of my cell, so here's my nuclei on the other side. we're going to start developing what is called a cell plate. And what that cell plate is going to do is it's going to help divide it. We're going to eventually build a new cell wall here, and then we'll have two different cells. So either way, this is both called cytokinesis for both. It's just keep in mind that because plants can't pinch in like that because there's a cell wall, they're simply going to build a new cell wall at this cell plate and then have two independent daughter cells from each other. That's pretty much it. So I know that was kind of quick, but that is the process of mitosis in a nutshell. I encourage you to kind of go back to the beginning, see if you can walk yourself through interphase, so G1, S, G2, and then talk about what goes on during prophase, metaphase, anaphase, telophase, and finally cytokinesis. If you feel like you got a good job and you can explain those, then I think you're in great shape. That's all there is really to it. I encourage you to go back and watch if you need to, but I hope you guys learned something, and thanks for watching.